Morning. Hi everyone. I'm Sornambiga, Assistant Professor in Microbiology at SNS College of Nursing. Welcome to the session on gram-positive bacteria and five-pillar concept followed by SNS institution. In this presentation, we will explore how to gram-positive bacteria helps to diagnosis in the disease condition and then the framework of learning, researching and clinical excellence to the topic. That gram-positive bacteria to a comprehensive framework to the healthcare excellence and that combination of the bacterial structure and identification strategies and treatment of the protocols. In the gram-positive bacteria, as we saw in the gram-negative bacteria, it is a thin peptidoglycan. It is a thick peptidoglycan cell wall to retaining the crystal violet strain during the gram staining process. In the gram staining process, we will already saw in that video that crossing them that appear in that distinctly purple color in that under the microscope. Here I am give the small picture to for your reference purposes. Unlikely, the gram-negative counterpart is the bacteria, lack of outer membrane. It is contain outer membrane. So in that common morphologies of the cocci, it is a spherical cell arranged in the cluster shape or else a chain shape. In the cluster shape, we can also like imagine is a grape-like a structure. The chain like a, it is also it's a chain like a structure also present in that. It is called streptococci. If it is in the cluster shape, is called as staphylococci. So that basically also in the rod shaped, it is that maybe occur in the single or as a pair or as chain in that shapes also they can able to we can able to see from that microscopical nature. After that, adhesion mechanism in the peptidoglycan, it is the main thing of to adhere in that human body. It is also in that thick peptidoglycan present in the outer layer of the bacteria. After that, it is a pili fimbriae. It extends from the cell surface to facilitating the attachment to the host tissues and the environmental surfaces. After that, the enzyme system, it is a convolently anchor the pilings of the cell wall. It is creating the stable adhesion of the structure in the essential to colonization formation. It is a adhesion mechanism is a critical and host colonization. It is a biofilm formation also. Whether the bacteria going to attach with that host, it is producing the biofilm. After it will going to attach with the help of the polypeptidoglycan. It is a water lovers, so they can able to attach anytime, anywhere by the way of air, water, and that atmospheric conditions also. Moving on to that, the gram-positive pathogens of the clinical infancies. Here I am giving for that 4 examples to your reference purposes. One is Staphylococcus aureus. It is a cause of spectrum. It is an abscess of life-threatening bacteria. We can able to identify by the way of gram staining method, and that MRSA strain present in the significant treatment for the challenges in the healthcare communities. Next is a Streptococcus pneumoniae. It is a pneumonia causing bacteria, pneumonial fever, pneumonial virus, like that pneumonial infection they can able to cause from this. Particularly it is affected from the children and then early stages of the children also, that immunocompromised patients also they can affected from the way of Streptococcus pneumoniae. It is a pneumo fever. Then moving on to the enterococci, it is a nosocomial pathogen by the way of UTI infection, bacteremia, or else it is similar like a fungal infection. So we cannot able to identify which one is a fungus and which one is a bacterial infection. So the enterococci almost similar to the fungal infection, but it's not at all the fungus infection. And then Clostridiums, it is known as the Clostridium tetani, it is a Clostridium difficile. It is a diarrhea causing infection. In this cases, that antibiotic associated with that diarrhea in the healthcare burden we can able to identify by the way of gram staining methods. And the diagnosis in that laboratory identification that I mentioned again, it is a gram staining. It is a crystal violet dye retaining by the thick peptidoglycan layer. So we can able to see the bacteria by the way of it is a violet color or else a purple color in the nature of the under the microscope. And the next one is a coagulation test. It is differentiate the Staphylococcus aureus in the coagulase positive, that another than that coagulase negative test. It is a one type of biochemical test. It is a coagulation test. And then culture analysis, by the way of qualitative morphology, we can able to identify that blood agar provide in that identification clues. After that, biochemical test is the specific confirmation identification by the way of antimicrobial susceptibility test. We can identify by the way of diagnosis. And the treatment and challenges. In the era we have to 1940s, penicillin is a major treatment we can follow, and 1960s is the first resistant we can methicillin we can give the patient by the beta lactamase resistant antibiotics. And that MRSA epidemic also we can in 1980s. Now we have to follow so many therapies for the strategies to kill the bacteria in that critical challenges. After that, integrated with the 5 pillars, it is a strategic implementation of the SNS institution. It is a structure and adhesion of the patient and rapid diagnosis, innovation and investment, and the healthcare treatment. Is these all the treatment we had to follow in these cases? To the global impact, the gram-positive bacteria is among the leading causes of the infection worldwide to affect the millions of animals across the institutions. Like that 5 pillar approach is the easily identify or the easily uh, observe or easily understand the topic in that cases of that. Then we have to follow that next video. Thanks for watching. Thank you.